Hi, we're here with Alex Pollen. He's an associate professor at UCSF and a member of the Kavli Institute for Fundamental Neuroscience, and his graduate student, Matthew Schmitz, who is the first author on this great paper that came out in Nature today called The Development and Evolution of Inhibitory Neurons in Primate Cerebrum. It's also an open access paper, so that's awesome. So everyone should download the PDF after. So welcome.
Oh, thank you. Thanks for having us.
So, broadly speaking, your paper looks a bit at what makes, primate brains, right, which includes humans, special. So primate brains, as we know, are larger than rodent brains, right, have more neurons, have an expanded cortex, and potentially new cell types. So how did you go about investigating this in your paper?
Well, we're really interested in these broad questions of how brains evolve and the extent to which brain evolution is driven by quantitative changes simply in cell number and brain region size, or if there are qualitative changes and new parts in the primate brain. Fathers of neuroscience has been inhibitory neurons. There's been this appreciation since Cahal that there's an increased morphological diversity of inhibitory neurons, especially in the human brain. Cahal went to suggest that this involved qualitative changes linked to the functional superiority of the human brain. And many of these changes in the adult brain are manifest early in development. And so we want to ask to what extent are the initial classes of inhibitory neurons that are first specified early in development conserved between primates and rodents over this long evolutionary distance. And that's where Matthew stepped in.
Yeah. So I guess to say a little bit more about what an initial class is, the idea is essentially the brain is formed sort of as a sheet of cells that are dividing, these neural stem cells. As soon as a neuron is produced, it sort of stops dividing and becomes what it's going to be for the rest of its life. But it has a lot of maturation to do. And so we weren't exactly expecting this, but we found that this initial class, this state that neurons begin in, there's a discrete number of them. There's I think 16-ish across the cerebrum. And so we were able to sort of put together a taxonomy of all of sort of what the inhibitory neurons are initially made up of.
Yeah, it's this really exciting time where there's this emergence of adult brain atlases. Instead of just looking at a few morphological or physiological features of cell types, we can look at expression of thousands of genes in thousands or hundreds of thousands of cells and group these cells according to the dimensional properties. And one of the patterns that's emerging is that there's a greater apparent diversity of cell types in the adult brain compared with the developing brain. And so early in brain development, we're not seeing the same extent of cell types, but there haven't been really comprehensive developmental brain atlases yet. And as Matthew was doing this work, he could find these discrete initial classes that we can see on the left slide of these maps. These maps represent, each dot represents a single cell, and they're grouped by transcriptional similarity. And we find about 16 initial classes of inhibitory neurons that are later partitioned into one or many terminal classes as these cells mature through processes of post-mitotic phase refinement. So we had this discrete set. There's not a one-to-one correspondence between cells as they're born and what they become later in development. And so we generated this comprehensive atlas of primate development that Matthew then compared to mouse, specifically for inhibitory neurons of the cerebrum. And then Matthew was able to put together a taxonomy where he linked these initial classes to their presumed terminal classes. And I think you might even have a picture of that here.
Yeah, we went through and we gave everything a standardized name so that hopefully people, as they go forward and recognize these things in their own data, they'll be able to tie it back to what we've done and what people before us have done.
And we tried to follow this nomenclature from the Allen Institute, from Jeremy Miller and Amy Bernard, who's now at Kavli, of naming cells based on evolutionarily conserved and likely cell intrinsic gene networks that define these cells. And by generating a comparative taxonomy with human and mouse, we could find these core genes that were shared across species and maybe more universal handles for naming these cell types. And then we also named them based on their presumed birthplace. So now we can organize this diversity around the birthplace, the presumed birth date based on the earliest appearance of the newborn neurons, and then these initial classes. And then Matthew used a lot of kind of the latest computational tools to link these initial classes to emerging adult brain atlases and make these predictions, many of which are consistent with lineage tracing studies, but some of them are novel or potentially even controversial assertions where we might not have expected an initial class to make multiple cell types that we have listed here. And, you dove into one of these, right? This eccentric spiny neurons that were recently discovered.
Yep. Right. I mean, that is one of the.
Oh yeah, that's another example you have up there. But there was this surprising cell type. Before we get to the evolving cell types, there was a surprising example of a cell type recently discovered in the striatum from R.P. Saunders and Steve McCarroll's lab, called an eccentric spiny neuron. And it was presumed this was a derivative of more canonical medium spiny neuron classes. And one thing Matthew found was the same initial class. Yeah, I don't think we sent you a slide for this one, but Matthew found that the same initial class that produces these eccentric spiny neurons also produces some neurons bound for the amygdala, some neurons bound for the olfactory bulb, and a very rare neuron that Matthew found in superficial white matter of the cortex. And so this was an example of this initial class concept where a relatively homogeneous initial class can send neurons that go off to many places and were previously recognized as many distinct adult cell types, but we're able to identify new coherence here from a developmental perspective.
Yeah, so I love that you were able to combine, sort of existing mouse data sets, generate new data, and really look, to create this new insight into brain development and evolution. And it sounds like there are different sort of potential mechanisms for some of this diversity in evolution. So could you speak to maybe one or two of the other examples that we might have slides for?
Yeah.
Sorry about that. Absolutely. I'm just excited about the initial concept as well. But the big question was, were all these cell types conserved? And Matthew found these two kind of contrasting mechanisms by which cell types may qualitatively change in the primate brain.
Yeah, I will say that it was very cool to see that almost all of them are conserved. Because you think about like the mouse brain and the human brain, and there must be something like dramatically different about them, but they're pretty much made of the same things. And so like the sort of having an evolutionarily novel cell type is extremely rare. But we were really lucky in looking at our data to sort of find, one of those examples is of a totally new cell type, and those are the striatal interneurons. These TAC3 positive cells that Fenna found last year. But she found them in the adult brain. And so we were very interested to see if they were sort of manifest in development or if they were something that emerged only after cells had matured. And it turned out the answer was that this totally novel TAC3 positive striatal interneuron is in fact found in the developing brain of primates and not rodents or any other mammals. And it also has like a very distinct sort of molecular profile where it has receptors for different neurotransmitters than the original cell type does. And so where all other mammals seem to, or at least rodents, they have this one striatal interneuron type that has receptors. Now primates have two, and they have sort of seemingly different roles in how they might function in a circuit.
Yeah, the gene expression data suggested that even very early in development, these cells already express different neuropeptides and neurotransmitter receptors, and also different transcription factors. And so it might be that the developmental emergence of this qualitatively new cell type is consistent with some of the evolution, where new transcription factors access different parts of the genome. So even though all cells share the same genome, these cells can change through genetic individuation of different regulatory elements responding to these new transcription factors or this new combination of factors, leading to the expression of functionally different gene networks. And we think this is an exciting test case where we can look at how that unrolls early in development. And Matthew could already find these gene expression signatures in dividing cells in the subventricular zone, as well as newborn neurons in the striatum.
Very cool. So, we have a question. What did you find most surprising?
Well, I mean, I guess what I found most surprising is probably the sort of other example of this cell type evolution that we were looking at. And so that sort of started with because that is expanded in humans and all primates compared to rodents. And we sort of expected to see, like everywhere else, that everything was sort of one-to-one relationships between primates and rodents. But we actually did not see that. We saw this sort of mystery inhibitory neuron in the cortex in our data. And because we sort of knew it was there, but we didn't know where it was, because using this single cell RNA-seq technique, you sort of lose where all the cells came from exactly. We sort of embarked on this "Where's Waldo" expedition, looking for this cell. I mean, to sort of sum it up, we sort of came from two directions. One was we looked into the tissue at these cryosections because we had a profile of what this cell should look like in terms of its molecular profile. We were able to track it down where it was, and it turns out it's in the deep white matter of the front lobe of the cortex, which is really was part of your cortex. It's known sort of as the decider, the prefrontal cortex is, and also it plans motor actions and stuff like that. And so it was really cool to find out where it was. And then the second cool thing is that when we were actually comparing between the mouse and the primate in just the data, we found that it sort of does have an analog in that these cells in the primate cortex looked essentially the same as these neurons that migrate a long distance into the olfactory bulb. And so we sort of followed these streams, and they all kind of go back to, there's a great image that we have of, they all come from the same place, but it seems that these olfactory bulb inhibitory neurons are, they're not only going to the olfactory bulb in primates, but they're sort of migrating in a large sort of chain migration stream into the cortex. And yeah, you can see that here, where the rostral migratory stream, RMS, is going to the olfactory bulb. But you can see that there are these other highways, which are the arc that Mercedes Paredes has really illuminated with her work. And then this new sort of straight into the prefrontal cortex and anterior cingulate cortex, the arc ACC. And that was
very cool.
What I think the coolest thing.
Yeah, we think it's so interesting in these cells migrating to the olfactory bulb are among the latest born neurons in the telencephalon, and then they're involved in maintaining plasticity in the adult olfactory bulb in rodents. And so we find it very intriguing that these late born neurons regulating plasticity are redistributed en masse to the latest maturing regions of the primate frontal lobe, the white matter that takes multiple decades of life to fully mature. And we're really interested in the functional implications. And in the course of following these neurons, Matthew also found another cell type on the previous slide that hadn't been identified one, that kind of forms a wreath around the striatum. It is molecularly looks like another type of olfactory bulb neuron that, in the olfactory bulb at least, produces dopamine. This cell expresses some of the machinery to make dopamine along the striatum. We don't know if that's what it's doing. And Matthew has named these striatum loriatum neurons for this wreath that they form. And we've also found them very sparsely in the claustrum, another region of the deep cortex that's involved in binding information between different cortical areas, and where it's been proposed that there might be sparse inhibitory neurons that help regulate this between regions of the claustrum. And what these findings give us are molecular tools where we can access these populations to study their function further. And we can also study whether they have a role in disease. And so these late migrating neurons to the white matter, it's known one of the more reproducible cell type composition associations in schizophrenia are these abnormal accumulations of neurons in the frontal lobe white matter. And with this increased molecular cell type resolution, we could go back to pathology tissue and ask whether these cell types are represented, and given the late migrator born earlier. And so we've put together this redistribution model, and Matthew calls it this reduce and reuse hypothesis, where the relative reduction of the olfactory bulb compared with the source progenitor zone for some of these inhibitory neurons, the dorsal lateral ganglionic eminence, the DLGE, that the relative reduction of the olfactory bulb compared to these progenitors may allow these cells to be co-opted for new functions in dramatically expanded areas of primate white matter or in the striatum. And that gets back to this theme that's more broadly in the lab, where the unequal scaling of brain regions as primate brains expanded. Not all regions changed equally, with olfactory bulb as maybe the most dramatic example. That this process of unequal scaling combined with new functional requirements may drive to this large brain cellular ecology, that cells exist in this new foreign, 1,000-fold larger brain in humans compared with rodents. And either new cell types are born, cells migrate to new locations, or existing cells have to learn new tricks to exist in this environment and adapt. And so instead of simply thinking about, as we have in our past studies, of how genetic and cellular changes might drive brain expansion, we're now thinking about the flip side of this, how brain expansion and reorganization puts pressure on cells and may also create vulnerable joints in the brain that have not completely co-adapted to the pressures of brain reorganization and represent trade-offs.
Yeah, no, thank you. I think this is fascinating, just the different sort of mechanisms for how cells can evolve. It's I'm excited to see what you guys publish next. Before we end, is there anything else you want to tell folks, something we missed, or say hi to your moms? I don't know.
Oh, definitely want to say hi to our moms. I now I can send this to her. Haven't seen her for so long due to the pandemic, but she's coming next week, so I'm really excited, Mom. But one of the things in terms of future directions is we'd like to know when these initial classes of neuron and represent their more molecular features of their terminal class, and how do these neurons make the decision of migrating to the olfactory bulb versus these new locations, and are there intrinsic changes already in this apparently homogeneous initial class, or are there extrinsic changes that guide their migration? And that's one of the things Matthew's currently following up on, is trying to trace the molecular changes in these neurons along their migratory routes to these diverse locations.
Exciting.
Yeah, thanks for talking to us. We're so excited to have the paper out, and tremendous work from Matthew spotting these patterns in the computational data and the histological data.
Yeah. No, it's a great story, and yeah, I was glad to be able to talk to you about it and share it with other people. So
yeah, and before we go, I want to highlight the other UCSF collaborators on the. We were extremely lucky to team up with Mercedes Paredes, who initially described the arc migratory stream with Arturo Alvarez-Bulla, and to add another chapter to that in terms of one of the major sources of cell types and the kind of reuse of this chain migration strategy used in rodents to make these long distances to the olfactory bulb, but also reused here in a number of settings. And then we were lucky to team up with Bill Seeley at UCSF, who's an expert on the selective vulnerability of cell types, to find that these striatum loriatum neurons, they're not just something in rhesus macaque. We also could find them in the human brain, even at 88 years in the same location. So they're also a human feature that persists throughout life, but one that you couldn't study in a rodent model. And this speaks to what are the limitations of rodent models as this major resource for biomedical studies. And then we're also lucky to team up with Jimmy Yee, who co-advised Matthew on the computational analysis and is a real leader in single-cell genomics, and with Tom Nowakowski, a real expert on brain development and cortical development, who's also recently described some other potential origins of inhibitory neurons in the cortex.
Great. Well, thank you guys again for joining us.
You're welcome. Thank you. Thank everyone for listening.
Thank you.
Signing off.
All right, bye.
Great.
Bye.